Polymerase chain reaction, or PCR, is an indispensable tool in biochemistry and molecular biology that allows researchers to amplify selected sequences of DNA from a single copy to millions of copies. PCR was developed in the early 1980s by Kary Mullis, who shared the Nobel Prize in Chemistry in 1993 for his contributions to this technology. PCR relies on a series of heating and cooling reactions called thermocycles that allows for repeated DNA melting and enzymatic replication. A successful PCR reaction requires a DNA template, DNA primers, dNTPs, a magnesium-based buffer, and a heat-stable DNA polymerase, such as Taq polymerase. A sample of chromosomal DNA, also called genomic DNA, can serve as the DNA template or starting material for the reaction. In this demonstration, the DNA template or gene of interest is indicated in red. The DNA primers, which are short oligonucleotides, about 20 bases long, contain complementary sequences to the ends of the gene of interest. Taq polymerase is derived from a bacterium which grows in hot springs and deep-sea thermal vents. Taq polymerase is thus ideally suited for the thermocycling conditions of PCR. A PCR reaction is comprised of 20 to 35 repeating cycles of DNA replication. Each PCR cycle contains 3 steps: 1. Denaturation, 2. Annealing, and 3. Elongation. In the denaturation step, the temperature is increased to 95 degrees Celsius for 30 seconds to a minute. This enables the melting of the double-stranded DNA template. That is, the hydrogen bonds holding the alpha helix together are disrupted, and the template DNA becomes single-stranded. Following denaturation is the annealing step, in which the temperature is reduced to between 48 to 60 degrees Celsius. The exact temperature depends on the nature of the sequence being amplified. In this step, the DNA primers anneal or bond to the DNA template through hydrogen bond interactions of the complementary bases. Adenine, A, pairs with thymine, T, and cytosine, C, pairs with guanine, G. In the third step, the elongation phase, the temperature is raised to 72 degrees Celsius, the optimal temperature for Taq polymerase. Taq polymerase begins elongation by adding nucleotides to the 3' hydroxyl of each primer attached to the DNA strand. Here it is in more detail. The chain elongation reaction is an attack by the 3' hydroxyl group on the innermost phosphorus atom of the incoming nucleotide. Pyrophosphate is released and subsequently hydrolyzed to yield 2 orthophosphate ions. A phosphodiester bridge connects the growing strand to the latest nucleotide, which brings its own 3' hydroxyl group, ready for another reaction. After one complete cycle, there are 2 double-stranded copies of the target DNA. The denaturation, annealing, extension cycles are repeated a total of 20 to 35 times. Initially, each reaction cycle results in the exponential amplification of the target gene. During the exponential phase, the copies of the target sequence continue to double with each cycle, resulting in millions of copies of the target gene. During the later stages of PCR, the reaction levels off, as the DNA polymerase begins to lose activity and the dNTPs are consumed. After cycle 3 is finished, there are 8 copies of the double-stranded target sequence. Note that only 2 of the double-stranded copies consist of just the target fragment. The others also include flanking DNA regions. As the number of cycles increases, the products consist of a greater proportion of fragments with just the target DNA. After 4 cycles, half of the fragments consist of just target DNA, and half of the fragments also contain flanking DNA. Often, PCR conditions must be optimized to result in the amplification of the gene product. Contaminants within the purified template DNA can inhibit the DNA polymerase and cause the reaction to fail, or can lead to the spurious amplification of contaminant genes rather than the target gene,